Now, just follow along, see if you catch what I'm doing, and then we'll see if it is, in fact, how you read. So when we get started with one of these articles, by the way, when you download them, where do you open them? iBooks. I opened it and explained everything so I could annotate on it for you, but you don't need to do that. You can just open it in iBooks. If you want to open it and explain everything for the purposes of annotation, you can do that. Not a big deal. Yep. Or Notability for those of you who are more familiar, comfortable, whatever your choice is. First thing we do when we're reading one of these articles is we look at the journal that it was published in. We can always find that at the top of the paper. And then we ask ourselves, does it sound like a reputable resource? So does the International Journal of Antimicrobial Agents sound like a publication that we should trust when investigating disease-causing pathogens? God bless you. Is it like Time Magazine, People? It's not like any of those. Not even like Science Daily. And it's certainly not Wikipedia. So it sounds like a resource that we're pretty comfortable trusting. So then we move on from the journal to the title of the article. That's right here, and it's what drew us to the article originally, right? Antimicrobial efficacy of silver ions in combination with tea tree oil against Pseudomonas, Staphylococcus, and Candida. You see the words Pseudomonas, Staphylococcus, and Candida, and you're like, that's Latin, I don't know what that means. Yes, you do. It's Latin. It's italicized. The first word is a capital. The second word is lowercase. So what does that mean?
It's the genus and species.
Yeah, so it's a genus and species of an organism. Do you need to know which one? No. It doesn't matter for the purposes of us understanding the title. You just know that they're microbes. I can tell you, based on what I know about them, Pseudomonas is a protist, Staphylococcus is bacteria, and Candida is yeast. So, Protista, bacteria, specifically eubacteria, and yeast is, which one? Yeast. Fungi? Protista, eubacteria, fungi. What do you know about those three organisms based on what your knowledge of classification is? What are three of those three things? I can tell you some of the others because there are 6 total. Yeah, they're 3 different kingdoms. So in this experiment, we're looking at 3 different kingdoms and how effective 2 substances combined are at killing members of those kingdoms. That's what we got out of the title. Is that relevant to my hypothetical project? Does it sound like what I was interested in yesterday? So we'll keep it still. After the title, we look at authors. Here, I don't expect you guys are going to know anything about the authors, but I will tell you or point something out about authors to you. Certain publications do not publish first names. You'll notice this is one of them. There's only last names here. Why?
Some of those people might not want people to lift them up and stuff.
We could think that it's for their privacy, but it turns out people who write this stuff want desperately to talk about it. So much so that they actually put their contact information down at the bottom of this page. They put their phone number and their email address. They're obsessed with talking about what they love.
Professional.
Why is it more professional?
Because they have to use names, like, in hypothesis, like, naming stuff.
What can't we tell about these people?
If they're, like, doctors.
Can't tell if they're doctors.
Not if they're male or female.
Can't tell if they're male or female. That's why publications leave it off. There is still a huge bias in certain disciplines of science, either for or against males or females, depending on the area of science, depending on the country that the research is done in. So some publications choose to not publish it.
Why don't they have their qualifications?
Their qualifications, what you'll find here, are actually by department here. My guess is none of them say PhD because none of them are PhDs. The only way we have to evaluate whether or not we trust these people is to see what departments they've been doing their research in and the institution they've been researching at. So that's the next step. We look at the authors' names and where they came from. Now I'll tell you, if you can't see it, it says the University of Wolverhampton. Sounds made up, but it's in the UK. It's real. Truthfully, I didn't know anything about the University of Wolverhampton, so I looked it up earlier. The information I found explains why none of them have PhD or MD after their names, or MS, or any other letters. These are not lettered people. I'm guessing that these are either students who've done this project, or the reputation of the university, which was only established in 1992, so it's like about as old as you guys. The reputation of the university does not draw individuals who are lettered to work there. Not to insult the fine folks at the University of Wolverhampton, but how did the website say it diplomatically? In the UK, it is one of the universities known for its inclusion of all individuals in higher university education, including a high percentage of individuals returning to higher education after careers in other areas. So you may interpret that as you choose. Like I said, they said it very diplomatically, right? So we have to consider that as we're reading. We're like, okay, that's an interesting thing to note. The next thing we look at is the date. 2010. New or old? Kind of oldish. We look for a window of 5 years or newer. This is just cutting it. I mean, this is squeaking in. You'll want to make sure, general rule of thumb, if any of the articles you found yesterday are older than you, don't use it. Be suspicious of science older than you in this arena. It's not new, it's not modern. Next thing we do is read the introduction. The introduction is called the abstract here. I have a feeling you'll be really good at this. So I'm going to have you do it without me. Your goal here in reading the abstract is to identify the independent variable, the dependent variable, and the way they measured it. All of those things are said here. Independent variable, dependent variable, and the way that they measured it. For those of you who are like, I can't see that far, don't worry. If you go to Science Direct and your key search term is tea tree oil and silver ion, it'll be one of the first results. Read it, discuss with your partner. I bet they'll have all kinds of fabulous ideas about what the IV, DV, and what they measured were. Go ahead and take, till the clock says 55 to do that. Here's the sentence. Tea tree oil and silver ions were added alone and in combination to suspension cultures of Pseudomonas, Staphylococcus, and Candida albicans. What is that? One sentence is their whole experiment. That's their experimental design. Experimental design has to tell us an independent variable, right? Tea tree oil and silver were added. If you add it, if the experimenter does the adding, changing, or manipulating, that makes it the independent variable. So our independent variables here were tea tree oil added alone, silver added alone, so tea tree oil alone, silver alone, and tea tree oil and silver in combination. So 3 things. There's a fourth group, though. Fourth group who never gets mentioned because all good scientists include it. We never have to mention it.
The control group.
The control group. Who's our control group? Tea tree oil, silver, both. And our fourth is
None.
nothing at all. They're all four there. To a suspension culture. All that means is they took our three microbes, they put them in a little test tube with food and dissolved them in a liquid. So a suspension culture is a suspension. It's a state of matter we're referring to. It's a liquid thing. So they put them in a suspension culture, added those things, and then, what did they do? That just tells us independent variable and method. What's our dependent variable here? You might call it results.
The order of sensitivity.
The enhanced loss of viability and order of sensitivity. That was their dependent variable. Now you're like, it doesn't sound like a measurement to me. I don't know how to measure that. They tell us a little bit later. But we know that their conclusion they drew from it was this order of sensitivity, and the way that they determined that is by a measurement that sounds totally made up, the fractional lethal concentration index. Made up, right? You're like, that doesn't sound real. The fractional lethal concentration index sounds like what people say to sound smart. Turns out it is a measurement that they created for the purposes of this experiment. Made up. That's all that is. How many of you are like, that wasn't, honestly, it was not a particularly difficult abstract. If I went through and I looked at the words and I was like, okay, so this is just a science term, but I get it. They're doing the scientific method. There's an independent variable. There's a dependent variable. Not a big deal. Not so bad, right?
Actually, are we supposed to be, like, in our abstract, are we going to have to use, like, all these big words and stuff?
When you become an expert on your project, big words naturally come out. You don't have to try. They just...
So they can just make up their unit of measurement?
Well, I'll show you how they did it, because they'll show us later on a table. And it's actually legitimately, if you look at how they did it, you're like, oh, that makes sense.
Wouldn't adding things to those three cycles affect the experiment circuitry if they were alone? Like, wouldn't the food be another variable that changes how the treatment stays out?
So all of them would get the food. Does that make sense?
Yeah, but like, what if they were on their own? Wouldn't that be a different? Would that even be an option? Like each microbe on like their own?
They do test them all on their own.
Oh,
Does that make sense?
Yeah.
Like it's, one trial is those 4 treatments of Candida, and then one trial is those 4 treatments of Staphylococcus, and then one trial is those 4 treatments of Pseudomonas. We'll see that later. They don't make it clear here, but they make it clear in their pictures. So as we're looking at this, I have good news for you. If you made it through this, if you identified the independent variable, you identified the dependent variable, you're like, okay, this sounds like the dependent variable. I don't know quite how they measured it yet. It sounds made up, but okay. That's all of the actual reading you're going to do. I'm going to do exactly what you do when you're given a reading assignment from a teacher, and you can tell me I'm wrong. I don't want to accuse you all of sloth, but something tells me it starts something like this. Look at the title of the chapter, look at the headings of the sections, look at the first paragraph, actually read the first paragraph, and then skim the rest. And by skim the rest, I mean thumb through it and look at the pictures.
It's funny.
It's funny because it's true. Guess what? That's all we do here. We evaluated the top half, we read the first paragraph, and then I'm going to be like, is there something like on this next page worth looking at? You know what, all this down here looks like a whole lot of words, and I don't really care about words. I'm skimming them. We call them emergency words. In case of emergency, you may look at them, but don't do it unless it's an emergency. When is it an emergency? When you look at the table and you're like, the it doesn't make sense, then you can go to the emergency words to figure it out. But most of the time, the caption is plenty. So we just ignore all that. Like, those, ugh, I'm not reading that. Crazy lady? They're like pages long. So instead, I just go up to the top of the table. And I take a look at it. And the table simply tells me my 3 different microorganisms. This right here, when I see %V/V, it's a measure of concentration. They're just telling me for this organism, the concentration of tea tree oil that they used. Because remember, they said they gave them the minimal lethal dose. So this is what that is for Candida. And this is what it is for Staphylococcus, and this is what it is for Pseudomonas. That's it. And then they do the same thing for our silver. And then silver, we have to do weight by volume. We can't do volume by volume. This is just another way to measure concentration. So they told us in our trials what concentrations they used. That was kind of them, wasn't it? Now remember I said that they were going to show us that FC or FLCI thing, how they got it? All they did was took one of their measurements of concentration combined with that concentration of that particular agent alone and did a ratio. Is a ratio a legitimate comparison of two things when trying to compare their effectiveness together and alone to make a ratio? So that's what they did. So like, yeah, it sounded made up, but it makes sense why they would do it. Because the ratio is just a comparison. That's not even an interesting picture, though. That's just like a, here's the measurements we used.
Why wouldn't they just call it a ratio to like what they're doing?
I don't know. Because it sounds better when you give it a fancy name. How many of you thought they were smart because it had a fancy name? How many were like, yeah, you used a big word. I believed you. Snake oil salesman. They made it up. Don't believe everybody who uses big words. Truthfully, the ones I trust are the ones that you know big words, but they don't use them. They use words that they think everyone understands because they don't feel the need to prove anything to anybody. Trust those people. You'll meet some one day. Mr. DF. Oh my gosh. Do you know how badly right there I wish I had the DF laugh? Because that would have been like the perfect timing for it. Admit it. That laugh makes you laugh.
Wait, what's the? I've never.
You've never heard DF laugh?
Actually, maybe I have.
I'll say something punny next time he's
Can
around.
we get him in here? Because I don't remember.
Yeah, just call
Can I
him
make
up.
that laugh?
It's kind of like, actually, truthfully, Mr. Dickey has the most amazing way to make these like little 3D renderings of people. And he's going to make like little, he made a 3D rendering of Dold and he made a 3D rendering of Breen. He wants to do a 3D rendering of Mr. DF, but I suggested that we can only do it if we make it a bobblehead and it makes the DF laugh.
Yeah.
He has electronics students who can figure that out. Sounds like a final exam to me. We moved on to the next page. I don't know if you guys noticed that. I was like, that was a lot of words. Nobody likes words. Next. It's exactly what you do when you're doing your reading assignment. You pause, have a conversation via text message with friends, and then go back to the next picture. So we went back to the next picture. Is it still funny because it's true?
Yep.
It's like I've been doing this for a while or something. I dare say it's almost like I used to be one of you. Once. A while ago. I'm not going to talk about how long ago. Don't even think about it. You're thinking about asking. I can tell.
Don't say it wrong.
Don't do it.
Don't say it wrong.
Don't do it. So the next thing we do is find the next graph, picture, chart, table, whatever it is. Picture, we don't like words. Pictures, pictures, pictures. This particular picture, you know you like the x-axis because it's an x-axis you're familiar with. I know you're familiar with it because my kindergarten-aged niece is familiar with it. Time. You understand that concept, right? Is that a bad x-axis then? I know my kindergarten age niece understands that concept because she came out of school on St. Patrick's Day and informed us that days that they do fun things at school, she must not be there as long because the day goes so much faster. Of course, it was St. Patrick's Day, so they had like a St. Patrick's Day party, because what do you do in kindergarten other than like have parties, read books, and eat snacks? And, right?
Oh,
So
well, she said that.
she also informed me that leprechauns visited the school. I was like, oh, I don't know that I like where this is going. We forgot to pray. Remind me to do that when we're done here. DF distracted me. So she came out, she's like, leprechauns visited. And I'm like, how do you know? The toilet water was green. And I'm like,
Yeah.
what?
I'm like,
And she's like,
Leprechaun.
they used all of the toilets in the school because all of the toilet water was green. Their teachers went to every toilet and dyed the water green so the kids would think that a leprechaun peed.
Oh, yeah, it goes.
That, so you're all aware,
Do a whole
is
nother
why
one.
I don't teach grade school.
I want to do that.
I'm not dyeing toilets. I love you. You're adorable. You want green toilet water? Go get some green food dye and do it yourself. God bless Amelia Bedelia.
Footprints really,
No, no.
thing to do.
Apparently, we dyed the toilet water green now. I suggested to my sister she might want to evaluate her choice of school.
Couldn't they just made like
No, like you know how like there were green footprints on like the ceiling when you came in. You were like, how'd they get up there in like kindergarten? And you were so freaked out because they were always on the ceiling.
We used to have to leave our shoes out in front of
St.
our
Nicholas
locker.
Day, right?
no, no. Hey, that's a Nicholas Day.
Listen, I'm okay with St. Nicholas visiting on St. Nicholas Day. Leprechauns on St. Patrick's Day. I might have a moral issue. This was like telling the kids that they rode their unicorns to town.
Didn't they?
Like, clearly.
Oh, yeah.
I don't know. The only thing on this chart, graph, figure, table, whatever we want to call this picture, that might confuse you is this. And I'll tell you, truthfully, it's not actually all that confusing. We can compare it to how many leprechauns peed. If the toilet water is only a little bit green, how many leprechauns peed?
Not that many.
Oh, not.
Just one leprechaun. But if it's very green, how many leprechauns peed?
Multiple.
Multiple
or it could
leprechauns
be
peed.
one.
We could probably make a graph that associates the number of times a leprechaun peed with the depth of green in the toilet water, right? That's all this actually is. All this is here is a measurement of how many of that particular microorganism there is in suspension, in liquid, right? Based on the amount of light that can pass through the liquid. Less light passes through, the more microorganisms there are. We make a logarithmic scale based on the amount of light that can pass through it and the number of organisms. It's totally normal in microbiology to use that measurement. That's all it means. Looks fancy. You'll learn it your freshman year of undergrad. If you go into any science, you'll do it in lab yourself. Not anything super complex. Again, which is why I'm betting, based on this particular experiment, that the fact there were no letters after their names, that's why there were no letters after their names, because this was probably something done by undergraduates. So is that scale really that bad? It looks bad, but they just put it in a machine that shines some light through it. A computer. They didn't even have to do the math. That's the worst part. A computer measured the amount of light and then told them how many microorganisms there were in the water based on the amount of light. That's it. For Candida, these are our 4 trials. So one was tea tree oil alone, one is both of them together, one is silver alone, one's the control group. So we put in our concentration of tea tree oil, and then we watched what happened over time by shining light through it over time. After 2.5 hours, but before 24, the break in the graph indicates that they didn't want to include all 24 hours. what happens to the concentration of organisms?
Dunzo.
If you were sick, would you want to wait that long for a response? When you're sick, 2.5 hours is a long time. How about the control group? It doesn't change at all, right? Pretty much stays the same. Is there like some kind of like mathematical calculation we can do to determine how quickly things changed?
So,
Yeah, we're going to do slope on this. That's all they'll do. And then they can tell us how effective it was based on how quickly it worked. Slope, that's it. Here, the squares being tea tree oil and silver. Most effective curve, right? Fastest acting, lowest concentration, most effective curve. Not zero, mind you. Didn't kill anything. Like it killed some, but it wasn't like a wholesale wiping out clean. It was just decrease in the population that's viable. Ideally, in a human being, if you decrease the population, then the human being's immune system can take over and finish the job, is what we're aiming for here. Was that picture really that bad then? Was it that bad when we looked at it initially?
Yeah.
Yeah. Until we compared the one axis to leprechaun pee, and then they're like, we got this. Guess what they do? Below. Same thing for Staphylococcus. Same thing for Pseudomonas. Make graphs, determine the slope. Now we have, they give them fancy names, time-kill curves. I call it a graph. They call it a time-kill curve. It's a graph. You know what we're doing with the rest of these words? Yeah, what do we call them?
Emergency.
Emergency words. Was there an emergency? Sweet, we can ignore them. Next. I bet you guys didn't know. It didn't take that long to read. But you've gotten everything you needed to out of this experiment, haven't you? You know the results. You know the independent variable. You know how they measured it. This top chart is going to tell us the minimum inhibitory concentration, so the smallest amount of tea tree oil we can use, and the minimum lethal concentration. So how much inhibits versus how much kills. That's just a results. It's a summary graph. It took all that data from those previous graphs, determined the amount or concentrations that were best to apply or administer, and we're done. One other thing worth pointing out in the emergency words, though, is this. Competing interests, none declared. We see this at the end of articles just so they tell us that they were not paid by anyone who manufactures tea tree oil or anyone who manufactures substances that contain tea tree oil and silver to do their research. It's just so we know they're not biased. They say it at the end. They also give us any ethical approvals. In other words, that IRB I told you guys, if it had to go through an IRB, that information would be here. They didn't have to because they weren't testing people, they weren't torturing animals, they weren't doing Nazi experiments, so they didn't have to say any of that. Congratulations, you just read something intended for doctors, PhDs, graduate students, and it wasn't that bad. Can you do it on your own? I'm not going to lie to you. I respect you enough to say it will take you longer than it took us here. That's okay, because you'll have time in class to work on that. Your goal is to have like 6 articles, so when you encounter 3 that are bad that you don't want, you can just chuck them. You can ignore them. If you encounter ones with like calculus that you can't, just be like, oh, that's just the mean. That's okay. You don't have to read that then. You have some backups. So your goal is to find 6 articles for next Wednesday, because Thursday, Friday, so you'll have 2 full A schedules to read. The whole A schedule. That's going to be the goal. So I want you to be prepared for that. Have what you need. I'll have a form for you to fill out while you're reading, so it'll be more helpful to keep track of independent variable, dependent variable, and methods. That's it. Prayer, I forgot to do prayer.